Hello, I'm Richard Rosen, and today I'd like to discuss quantification and dynamic characterization of intermittent retinal capillary perfusion in sickle cell disease using serial OCT angiography and adaptive optics angiography. First, I want to acknowledge the many members of my team who made this presentation possible. These are our disclosures. These are our sources of funding. Sickle cell disease is the most common inherited blood disorder. It's caused by a single point mutation in the beta hemoglobin gene. In some areas of Africa, it affects 1% of all babies. In the US, it affects more than 100,000 people. Under deoxygenated conditions, sickle hemoglobin self-assembles into long biopolymer fibers within the red blood cells that increase cell stiffness and blood viscosity. Here is a demonstration of red blood cells undergoing sickling under hypoxic conditions. Notice the variety of distortions in the cell body shape. Unlike normal red blood cells, which are deformable, these sickled cells adhere to each other and often get jammed at vessel bifurcations, resulting in focal occlusions. OCT angiography has enabled quantitative analysis of sickle cell retinopathy. Previously, we demonstrated how capillary density could distinguish proliferative and non-proliferative sickle retinopathy from healthy controls. Past studies have relied on single OCTA scans. Given the dynamic nature of vaso-occlusion in sickle cell disease, we wanted to see if more information was revealed from serial OCTA scans. On the left, you see a 10-scan sequence of OCTA imaging of a sickle cell patient. Some segments appear to flicker, suggesting intermittent flow. Adaptive optics scanning light ophthalmoscopy on the right reveals the blood flow events producing this flickering. Comparing OCTA scans to detect differences is very difficult. Even using capillary density maps, differences are not easily seen. We developed an intermittent perfusion map to highlight fluctuating capillary segments. This intermittent flow map shows which vessels reperfused at 1 hour and which vessels became non-perfused. The purpose of our study was to investigate this quantitative OCTA metric of intermittent retinal capillary perfusion and to examine the dynamics of these flow events using adaptive optics SLO angiography. 13 sickle cell patients and 14 healthy controls were imaged at baseline and 1 hour follow-up. 10 sequential 3x3 OCTA scans were obtained at the parafovea and 9 degrees temporally eccentric during each session. Scans were spatially registered and averaged at each retinal location. A normative reference of healthy controls was created using 10-scan averages of the full vascular OCTA slabs with non-capillary blood vessels removed. The two time points were then subtracted to determine the difference in pixel intensity. The in-between session difference in pixel intensity was then analyzed using the Bland-Altman plot. Limits of agreement of 0.1 and 99.99 percentiles was computed, generating segments considered re-perfused and non-perfused respectively. The normal curve was then applied to the sickle cell patients. Vessels which fell outside the norm were then highlighted on the map. Here is the first patient example. You can see multiple vessels outlined with intermittent flow. Here are maps of other patients with a variety of ages and genetic types. Box plots demonstrate that there was no statistically significant difference between the percent areas of non-perfusion and re-perfusion. However, between session intermittent perfusion index comparisons revealed a statistical difference between controls and sickle patients, both on and off hydroxyurea at both locations. We also looked at within-session intermittent capillary perfusion events. In this sickle cell patient on hydroxyurea treatment, the yellow arrows indicate 2 capillary segments with intermittent capillary perfusion. These within-session events were seen in all sickle cell patients and 1/3 of the 12 controls. Here's a 31-year-old patient prior to initiation of hydroxyurea therapy. On the right, you can see the effect of 2 months of starting treatment. The intermittent flow maps highlight the changes pre and post-treatment and quantify the difference. There's a 5 times reduction in intermittent flow events seen with this method. Looking at small regions of interest from the same patient using adaptive optics, we were able to see a congested rouleau which had its flow restored on treatment. Here's another congested rouleau which leaves single RBCs suspended in a capillary segment. After initiation of treatment, the flow in the segment is restored. In another quadrant, you can see sludging of a long segment along the FAZ border, which is then restored following treatment. And finally, here's an example of a congested rouleau blocking flow in the long segment. Following initiation of treatment, there's brisk flow. In summary, serial OCTA and intermittent flow maps reveal vascular perfusion changes in sickle cell not seen on single scans, highlighting differences from controls. Quantification of perfusion disturbances provides a metric of vaso-occlusive burden, which may be helpful for prognosis and evaluation of treatment response. Adaptive optics SLO imaging revealed details of abnormal red blood cell flow causing the intermittent perfusion and non-perfusion seen in the OCTA. Combining OCTA and adaptive optics imaging provides a clearer picture of the vascular anomalies of sickle cell disease, which may be helpful for evaluating new therapeutic approaches. Thank you for your attention.